welcome to Biology My Passion. I am Soumya Harikrishna. We have been dealing with the chapter Molecular Basis of Inheritance. Now we are exploring central dogma. We discussed DNA replication, transcription, genetic code, and now we are dealing with mutation. What are mutations? You know, DNA has a long sequence of nucleotides. So there can be some variations happening to those nucleotides. So if there occurs variations or alterations in the DNA sequence, then it is called a mutation. We know if the DNA sequence changes, it will change the genotype of the organism. If the genotype changes, phenotype also will change. So we can say that mutation can lead to variation. There is one more phenomenon which can change or cause variation. What is that? Recombination. We know DNA is a long strand of nucleotide, polynucleotide chain. A part of the DNA coding for RNA is called a gene. Now this gene, once it is coiling to form a chromosome, it is condensed to form a chromosome. So one helix or DNA double helix is making a very condensed form of chromatid here, right? So if a part of this chromatid is missing, that is called a deletion, some part is being deleted, or if a part is duplicated, or instead of duplication maybe there is inserted, another part is inserted, all these can lead to changes in the chromosome. All these changes happening to these chromosomes are called chromosomal aberrations, or we can say chromosomal abnormalities. Whereas sometimes the changes can happen within a gene also, because this gene is ultimately coming within the chromosome. But sometimes the change can only be inside the gene. So it can occur in the gene level also. So we can say that types of mutation can be intragenic or intergenic. Intragenic means what? Within a gene. So within a gene sequence, if there occurs a change, that is called a intragenic mutation. So if you know that the gene sequences are coding for proteins or ultimately enzymes, if a change occurs to that sequence, definitely it will alter the enzymatic activity of that particular gene expression. So that will alter the physiology of the person or the organism. Whereas if this is happening to a long stretch of DNA, then we call it as intergenic. That is mainly the chromosomal changes. So if you see the types of mutation, as we mentioned, chromosomal mutation can be there. That means a part of the chromosome can be deleted or added extra. The same way it can happen to the gene level, then it is called a gene mutation, or gene mutation is otherwise called a point mutation. Okay, so chromosomal mutation can occur in two ways. Sometimes the number of the chromosome may change. The total number, there can be a variation. The same way, sometimes it can affect the structure of a chromosome also. Okay, so the chromosomal mutation can be based on the number of chromosome changes or the structure of the chromosome. Now, if you see the number of chromosomes, we can have two types of chromosomal aberrations or mutations. First is called a euploidy and second is aneuploidy. Euploidy. Ploidy means what? The set of chromosomes that we possess. We know that being human beings, our chromosome number is 23. Different organisms have different number of chromosomes. So number of chromosomes is specific for a particular species. So we have 23 chromosomes, that is our single set or haploid, but we are diploid organisms. So we have one more homologous pair of these 23 chromosomes. So we have total 46 chromosomes. So we call it as 2n. 2n means what? Diploid condition. Our ploidy is 2 because every chromosome we have 2, right? A pair of homologous chromosomes we have. So that is normal for us, diploidy. So from the diploidy, these two are variations. So if you say euploidy, the whole set of chromosome will be either less or more. Not 1 or 2. The 23, the whole set will be more or less. For example, if there is less than the diploid number. We know diploid number is 46, 2n. If there is one set less, so from 1 to n, one set less means what? N. Okay. So that condition what do we call? Haploid, or it is also called monoploidy. But again we cannot reduce because one more set gone means no chromosome, so the organism is not there. So from the diploid condition, if one set is missing and only one set is available, that is called a monoploidy or haploid condition. Whereas on the other side, increasing. So if there are 3 or more sets of chromosomes, then we call it as polyploidy. Poly means what? Many, right? So sometimes it can be 3 sets of the chromosome. For example, if we have a chromosome set of 3 chromosomes, A, B, C, 3 chromosomes are there. It's repeating 2 times. This is diploid condition. But if it is repeating 3 times, then we call it as triploid. If it is again repeating, then it becomes tetraploid. One more time, it becomes pentaploid, then hexaploid, septaploid. It goes on like that. Okay. So that's about the change in the number. So if the whole set of chromosome is less than the actual diploid number, it is haploid condition. If it is more than the diploid condition, it is polyploid, and depending on the number, the names also vary. So if we look at aneuploidy, aneuploidy can be due to either reduction in the number or addition of the number. Here it's not the total set missing or adding. 1 or 2 chromosomes or 3, 4 may be added or deleted, not the entire set. That is called the aneuploidy. So reduction, first one, monosomy. Monosomy means what? Mono means what? 1. So 2n minus 1, our total number, suppose it is 46. From that only 1 is missing. That is called a monosomy. That is, suppose we have ABC one set. Next one only AB there. C is missing. So 1 is less than the total number. That is called a monosomy. Then next is double monosomy. Here 2 chromosomes are missing from the total. 2 different types like A, B, C, 3 different chromosomes. But next type, its homologous pairs are missing for B and C. So A, B, C, A only. B and C, their homologous pairs are missing. Or it can be nullisomy. Nullisomy means 2n minus 2. Here I wrote minus 1, but here I wrote minus 2. Both are same, right? The reason is, here it is 2 different chromosomes are missing. Here the same pair is missing. That is ABC, ABC it should be, but here only AB, AB. So the C, the complete homologous pair is missing. That's why now only 2 are there. That is called a nullisomy, 2n minus 2. Now let us see what happens if the number is added. So in addition, the first case is trisomy. Trisomy means what? The total number plus 1 extra. That we are learning in the case of Down syndrome. ABC, ABC, that's what we need, but 1 A extra K. Second case, double trisomy means 2 different chromosomes are being added up. That is 2n plus 1 plus 1. That is ABC, ABC. That is our diploid. Then 1 A is extra, another B is extra. 2 different chromosomes are extra. Whereas tetrasomy, here again 2n plus 1 and 2n plus 2 both are equivalent, but 2n plus 2 indicates 1 homologous pair is being added. Similar chromosomes are added. ABC, ABC and 2A added. Okay, so that's about the number. So whole set is added or deleted. That is called the euploidy. And we know the polyploidy that we use in inducing seedless fruits in plants by using a hormone called auxin. The same way, aneuploidy means a few chromosomes, 1 or 2 or 3 or 4 chromosomes, less or more than the total number, not the entire set. If we see the second type, that is based on the change in the structure of chromosome, again it can be due to 2 reasons. First one, a part here, number is not changing, total number is not changing, a part of a chromosome is lost or deleted, or there is a deficiency of a part is missing from a chromosome. Suppose this is a chromosome, from this one part is missing. The same way, It can be addition also. This is a chromosome, one part is being added. It can be the repetition of the same or added. Okay, it can be duplication, addition or repetition. Apart from this, there is one more way this change can happen. Translocation. If two chromosomes are there, from this one part is joining with the other one. These are homologous chromosomes, suppose. From this a part will be added to this, making a new chromosome combination. The same way, inversion. Inversion means if there is a sequence of base pairs here, so in the linear order of the genes, a particular one may rotate 180 degree. So the linear, we are reading in a linear way, it is rotating 180 degree. So the order is changing, that is called a inversion. Now we will see these types with the help of diagrams. So here are the pictorial illustrations of what we just discussed. Here first one, let us take the original form of a chromosome with some genes A, B, C, D, E, F. That's the original form. Now a part of this chromosome is missing. Here the B is not here, that part is deleted. Okay, so that is a kind of abnormality that is due to deletion. Okay, then next case, what are you seeing? Observe this, A, B, B, C, D, E, F. So here it is not deleted, but it is duplicated. This part B is duplicated. Now next case, A, B, C, D, E, F is the normal one, but another piece G, H coming from somewhere else. Okay, so this is from another pair of chromosomes, exchange of parts between homologous chromosomes. So here the GH genes are coming and getting incorporated between genes B and C. So now the sequence becomes A, B, G, H, C, D. It's a new combination of chromosome. Then next is inversion. That is, in the linear order of the gene was A, B, C, D, E, F, but now it shifted or it rotated 180 degree. So now it is read A, C, B, D, E, F. So these are the different types of chromosomal aberrations. Now we will discuss the gene mutation or point mutation. Now when we come to gene mutation, it is otherwise called a point mutation because it's not happening to a long chromosome, or it is happening only within the gene, within a small sequence. Okay, so here actually genes are not changing, within the gene nucleotide sequences are changing. So that can be of two types. One is base pair change or switch. Means instead of one nucleotide, another nucleotide will be there. It's not missing or adding, but only one is replaced by the other. Suppose if I am writing a word cat. Okay, we know cat is an animal, and by mistake when I am writing, I make a spelling mistake. I write it as rat. Are they same animals? No, both are animals, but they are not same. So here what happens? One letter just changed, C replaced by R. So such kind of mutation can occur. It is called a base pair change or switch. Only one position base pair change can occur. But that can have effect. That is, amino acid coded by this particular one will change. So the total amino acid sequence in that protein chain will eventually change because of this mutation. Now next type is called a frame shift mutation. Frame shift mutation is due to deletion or insertion, sometimes missing or sometimes adding nucleotides. Insertion means few nucleotides may be appearing which were not actually there. Okay, so let us see one example. A sequence here, it's a DNA sequence given, cat, gat, cat, gat, cat. But when it was copied, here see, one error occurred. Instead of C, what came? T. Instead of cytosine, thymine came. So only one position there is a change. That is the base pair change or a switch. But this happens in a disease, genetic disorder called a sickle cell anemia. We know sickle cell anemia is a disease which changes the shape of our RBC. What is the reason for that? The hemoglobin contains alpha and beta chains. Okay. If you see the beta chain, it has a sequence of amino acids. In the sixth position, it should be glutamic acid. Glutamic acid is coded by GAG. But there occurs a mutation or a switch which becomes GUG instead of GAG. But GUG is not coding for glutamic acid, it is coding for valine. So in the sequence, at one position amino acid changing, that eventually changes the shape of the hemoglobin under oxygen tension. So these people are affected by a disease called sickle cell anemia. So you see how drastic the effect is. Even with a single switch, the change is drastic. But it may not be always the same. Its effects may vary. If we talk about the effects of different types of point mutations, that is base pair change, it can be silent mutation sometimes. What do you mean by silent mutation? There is no effect at all. For example, if the sequence was GCC, original sequence was GCC, which codes for amino acid alanine. But by mutation, it became GCA. The C became A. But still it is coding for alanine. So is there any effect? No, the mutation is not causing any effect. Mutation happened, but there is no effect. That is called a silent mutation. Hope you can correlate this with the verbal hypothesis I taught you previously. Okay. Now, second is missense. That is, only one base pair switch will change one amino acid in a long polypeptide. Just like we saw in the case of sickle cell anemia. So there can be effect. Then last is very drastic, nonsense mutation. Nonsense mutation means what? Suppose the original sequence was CAG. But due to mutation, it became UAG. But we know UAG is a stop codon. Now what happens? With that, the translation will stop or protein synthesis will stop. It will not continue. So the protein is not complete. So the effect won't be there in the body. Right? So these are the different effects of the base pair sequence or switch. Now let us discuss frame shift mutation due to deletion or insertion. In case of frame shift mutation, deletion or insertion, this is the example given in your NCERT. Suppose there is a sentence with the 3-letter words, that is Ram has a red cap. This sentence makes sense, that's why it's a sentence. Now let us see, in the first case, here first R is missing. But we can read only in 3-letter words. If there is a condition that only in 3-letter words we can read. So it becomes Ram has R gone. So we have to take 1 from here to make it 3. So EDC, AP. I don't know any words to read like that, EDC or AP. They are not making any sense. Okay, but up to this there is meaning. Ram has something, but we don't know what it is because that is not making sense. Next case, what happens? Ram has R and E, both are missing. So what is their D? From here 2 letters we have to take to read it in the frame of 3. So DCA, P. Ram has DCAP. Makes sense. After the point of deletion, it is not making any sense. Up to that, it's not a problem. Ram has. Now D also gone. Completely 3 nucleotides are gone. Cap. So again it is making a sense. Ram has cap. So here 1 or 2 nucleotides were deleted. So the reading frame is changing from the point of deletion. And when 3 nucleotides are removed, the whole sequence is removed and again it is making sense. Now let us see if we are adding a letter. Ram has one D is added. So from here we can take only R and E. D will go to the next one. So BRE, DCAP. No sense after has. That is after the point of addition. Now 2 letters are added. Ram has BI. Then we have to take only 1 R here because these 2 will be pushed to the next one. So it will go EDC with a join with C, then AP. Again no sense. Ram has 3 letters we added, BIG, red cap. Big red cap Ram has. Very good, it makes again sense. So if we are adding again, like deletion, if you are adding 1 or 2 nucleotides, it is changing the reading frame. Frame shift mutation or shifting the frame of reading. That's a frame shift mutation. But if it is a 3-letter nucleotide or 3 nucleotides together, again it can code for 1 amino acid. Okay, so what happens in these 2 cases? If it is 1 or 2, this will change the sequence of amino acid and it won't make sense. So the entire sequence of amino acids will change in the protein. Due to this drastic change, the protein may become non-functional. But if this is the case, one amino acid may be missing or one amino acid will be extra, that may not be so drastic like the other two. So this is called a frame shift mutation. Let us now talk about the effects of mutation on phenotype. Some mutations are very dangerous. They are called lethal mutations. Lethal means what? Killing. These mutations can lead to the death of the cell or the organism. So, if lethal mutation occurs, the cell will not live or the organism will not survive. Next is subvital. Means it is actually a threat to the life. Means it has got a lot of abnormalities, but still it survives, but chance of survival is less. Then supervital. Supervital means what? Giving some added advantage, or some advantages are there offered by this mutation. Under certain condition, there will be improvement of biological fitness of that particular organism. We learned mutation can lead to variation. Variations are in a way good for adjusting with the environment or evolution. The same way, some mutation can be supervital also, giving some biological fitness to the organisms. So, sometimes mutations are good. Even in case of breeding, we have to induce mutation in certain plants. Mutation can be induced. How can we induce mutation? Using certain substances. So those substances which are used for inducing mutation are called mutagens. Mutagens increase the frequency of occurrence of mutation. So mutagens can be of two types: chemical mutagens and physical mutagens. Chemical mutagens example is mustard gas. This is the first discovered chemical mutagen. Mustard gas, though we tell it as a gas, technically it is not a gas. It is sulfur mustard, which is commonly used as a chemical warfare. The same way, physical radiations can be of two types: ionizing radiations and non-ionizing radiations. What are ionizing radiations? The radiations with a higher wavelength have got high energy value. Since their energy value is high, they can disrupt the structure of atom and they can produce ions in that atomic structure, or it can leave a free radical there. Such high wavelength, high energy radiations are called ionizing radiations. Examples are X-rays, gamma rays, etc. Whereas non-ionizing means the ones which cannot cause ionization, like UV rays, radio waves, microwaves, etc. All these are examples of mutagens. Hope you enjoy learning with me. If you like my teaching, please like, share and subscribe to my channel, Biology My Passion.